Good afternoon, students. In the last lecture, we have discussed about the heterosis and hybrid vigor. Okay, heterosis means when the two homozygous plants having the different genetic constitution are crossed with each other, and the progeny produced it is superior than that of their parents. Then, in that case, that phenomenon is called as the heterosis. Okay, heterosis and the hybrid vigor. Okay, we have also discussed about the history of the heterosis and hybrid vigor, then also discussed about the types of the heterosis. Okay, there are two types of the heterosis. The first one is the true heterosis and another one is the pseudo heterosis. Then also we have discussed the genetic and the physiological causes which are responsible for the heterosis in the F1 progeny. Okay, then also we have discussed the utilization and limitations of the heterosis and the hybrid vigor in the self-pollinated crops, in the cross-pollinated crops, and also in the vegetatively propagated crops. Okay, we have discussed all the aspects of heterosis in the last lecture. Okay, and now in today's lecture we are moving towards our last plant breeding method which is used for the improvement of the crop, and that is the mutation breeding. Okay, there are four principal methods which are used worldwide for the improvement of the agricultural crops. Okay, four methods to improve the agricultural crops and to release the new superior variety are used worldwide. Okay, of this one we have discussed in detail the first method, domestication, plant introduction and acclimatization. Then second one, we have discussed in detail regarding the selection, and the third method is the hybridization. Okay, and this is the last method which we are going to discuss, and that is mutation breeding. Okay, mutation breeding. Here the first of all, regarding the mutation breeding, what is meant by the mutation breeding or the concept of the mutation breeding? Okay, before going to discuss about the mutation breeding, you should know the basic concept of the mutation. That means what is meant by the mutation. Okay, mutations, that is these are the sudden heritable changes in the organisms other than those which are arised due to the Mendelian segregation and the recombination. Okay, mutation, it can be defined as the sudden heritable changes in the organisms other than those due to Mendelian segregation and the recombination. Okay, the term mutation, it was firstly used by the De Vries in the 1900 for the first time. Okay, De Vries in the 1900 for the first time used the terminology mutation. Okay, the term mutation, it is derived from the Latin word mutere. Mutere means to change. Okay, the term mutation, it was firstly coined by the De Vries in the 1900 for the first time, and it was derived from the Latin word mutere, and mutere means to change. Okay, nowadays the term mutation, it is widely used to all types of hereditary changes. Okay, all types of hereditary changes resulting from the alternation in the number one, it may be the chromosomal mutation, that may be we that is also referred as the ploidy. Okay, here the by mistake I have written the mutation. The word, the appropriate word should be the chromosome number. Okay, resulting from alternation in the chromosomal number. Okay, that is we referred it as a ploidy. Okay, please here please here, that is the mutation, it is not the mutation, actually it is the number. Please read as a chromosome number, which is referred as the ploidy. Okay, then the second one, the chromosome structure, changes in the chromosome structure, these are also referred as the chromosomal aberrations. These are the another type of the mutations. Then the mutation, it may takes place in the individual gene. When it takes place in the individual gene, it is said to be the gene mutation. Okay, then the last one, the changes in the somatic parts. it is said to be the somatic or the cytoplasmic mutations. Okay, that is the term mutation. It is widely used for the hereditary changes which are resulted from the alternation in number one chromosome number, number two chromosome structure, number three individual gene, and number four the somatic parts. Okay, the alternation in chromosomal number, it is referred as the ploidy. Alternation in the chromosome structure, it is referred as the chromosome aberrations. Alternation in the individual gene, it is referred as the gene mutation. Okay, it is referred as the gene mutations. Gene mutations, these are also referred as the point mutations, and these are also called as the factorial mutations. Okay, the alternation in the individual gene is referred as the gene mutations. These are also called as the point mutations, and these are also referred as the factorial mutations. Okay, and the last one, the changes alternation in the somatic parts, these are referred as the somatic or the cytoplasmic mutation. Okay, and generally nowadays, here you can see in the, that is widely applied for the mutation, that is it is widely applied for the hereditary changes which are occurred mainly due to the alternation in chromosomal number, chromosome structure, or the individual gene or the somatic parts. But in restricted sense, the mutation, the term mutation, it is used for the invisible changes, used for invisible changes, whether they may be physical or the chemical within the individual genes. Okay, in restricted sense, the term mutation, it is used for the invisible changes, whether these are physical or the chemical within the individual genes, which are called as the individual gene mutations or the point mutations or the factorial mutations. And these individual gene mutations, these are having the considerable importance in the plant breeding to develop the new superior variety. Okay, of these all types of the mutation, the mutation which takes place in the individual gene, it is of the considerable importance in the plant breeding. Okay, here the mutations, these are of two types, that is the spontaneous mutations, means which occur naturally. Okay, and the another one is the induced mutations. Okay, these mutations or the sudden heritable changes, these can be artificially induced with the help of the different types of the mutagenic agents. Okay, different types of mutagenic agents are available, and by using these mutagenic agents, the mutations can be artificially induced in the organisms. Okay, later in the latter slide we will discuss the what is meant by the mutagenic agents and the different types of the mutagenic agents which are used in the mutation breeding. Okay, and here the creation of mutation at will and their utilization for the production of the new crop varieties is known as the mutation breeding. Okay, the mutations can be artificially induced with the help of the different types of the mutagenic agents, and the creation of mutation at will and their utilization for the production of the new crop varieties is known as the mutation breeding. Okay, when we compare this method with the other conventional methods like the plant introduction and the acclimatization, then selection, and then hybridization, then this method, it is of recent origin. Okay, it is a modern method of plant breeding. Okay, Mueller in 1927 and the Stadler in the 1928 gave the birth to the mutation breeding technique. Okay, it is of recent origin when we compare it with the other method. This method is a modern method. Mueller in the 1927 and Stadler in the 1928 gave birth to the mutation breeding technique. Okay, then this mutation breeding technique, it is considered as the magic tool for creating the new varieties. Okay, it is considered as a magic tool for creating the new varieties. Why it is considered as a magic tool? Because by using this method, the new variety is produced with the entirely new and the original characters. Okay, by using this technique in the crops, the new variety it is produced with the entirely new and the original characters, which is not possible in the other methods. Okay, why it is not possible in the other method, and why the variety which is produced by the mutation breeding, it is different from the variety which is produced by the other methods? Because for example, in the selection you can see the no new character is introduced, okay, or the no new gene is introduced in the variety. Okay, only the best genotypes or the best characters which are present in the population, that is, these are sorted. Okay, that means the characters which are present in their parents, these get expressed only in their progeny. Okay, in the conventional methods like the selection. While in the hybridization, what happens? The two pure lines having the different genetic constitution, these are crossed with each other. That means in the hybridization, the genetic constitution of the two pure lines, it is combined together only, okay, to form the new variety. But in the mutation breeding, what happens? The variety it is produced with the entirely new and the original characters. Okay, and that's why the mutation breeding technique, it is entirely, or we can say, completely different from the other conventional breeding techniques. Okay, because in this one, the variety in the variety, entirely new characters are introduced. That one we can say the entirely new genes are present in the variety, which is possible only in this method, and it is not possible in the other methods. Okay, that's why here we are saying the mutation breeding, it is the magic tool for creating the new variety with the entirely new and the original characters. Okay, with the new and the original characters. Okay, that is about the term mutation and the mutation breeding. Okay, here you can see just in the previous slide, I have told you that there are two types of mutations. Okay, the spontaneous mutations and the induced mutations. Okay, the what is the difference between the spontaneous and the induced mutations, and why the mutations are induced artificially in the crops? Okay, here in this slide we are going to discuss this reason. Okay, here the on the basis of the types of the basis of their origin, there are two types of the mutations. Okay, on the basis of types, on the basis of their origin, there are two types of mutations: spontaneous mutations or the induced mutations. Okay, spontaneous mutations, they arise automatically in nature. Okay, they arise automatically in nature when the organisms, these are subjected to the subjected to the different mutagenic agents like the electric current or the atomic rays or the injuries due to the certain types of the diseases, insect attacks, and the temperature. Okay, then these type of the mutations are called as the spontaneous mutations. Okay, spontaneous mutations which arise automatically in the nature. Okay, for example, just we are seeing the in this pandemic situation the different strains of the COVID virus. Okay, that virus it get mutated very fast. Okay, and it get mutated due to the different conditions like the different types these came in contact with the different types of chemicals, or the due to the certain type of the injuries, that is, these get mutated very fast. Okay, this type of the mutations we are calling them as a spontaneous mutation. Okay, spontaneous mutations arise automatically in the nature when the organisms came in contact with the electric current, atomic rays, injuries, diseases, insect attacks, and the temperature. Okay, these type of the spontaneous mutations, spontaneous, or we can say the natural mutations. Okay, that is, they remain arising constantly and continuously in the nature. Okay, the spontaneous mutations remain arising constantly and continuously in the nature, and these mutations are the basis of the all crop improvement by conventional breeding methods. Conventional breeding method, we have discussed the different artificial selection methods like the mass selection, pure line selection, and the also we have discussed the clonal selection and also the hybridization. Okay, that means these are spontaneous mutations. These are very important as these are the basis of a crop improvement by the conventional breeding methods. Okay, by the conventional breeding methods. But these are spontaneous mutations. These are very slow. Okay, spontaneous mutations, these are very slow. These are infrequent in nature. Okay, these are very slow. These are infrequent in nature. These are infrequent in nature, and that's why these are not sufficient to speed up the improvement of plant in accordance with the increasing human needs. Okay, as these are very slow and infrequent in nature, these are not in accordance with the increasing human needs. Okay, and these are not sufficient to speed up the improvement of plants or the improvement of the crops uh, in accordance with the increasing human needs. Okay, and that's why to speed up the crop improvement process, thus the this due to which the human beings are forced to induce the mutations artificially in the plants at their will. Okay, thus it forced the man to induce it artificially in the plants at their will, and that's why the mutation breeding technique was discovered to just speed up the method of crop improvement. Okay, and that is the difference between the spontaneous versus the induced mutations. Okay, that is the difference between the spontaneous versus the induced mutations. Then the different types of the mutagens, here you can see the radiations. Radiations, and number two, the different types of the chemicals, these are used for artificially inducing the mutation in the plants. Okay, artificially inducing the mutation in the plants. Here the first one, the radiation. What is the radiations? That is, these are the movement of energy given by the radioactive isotope treatment. Okay, movement of energy which is given by the radioactive isotope, that is we called it as a radiations, and the treatment of plants with the radiations to induce the mutation, that is called as the irradiation. Okay, that is called as the irradiation. Okay, and sometimes therefore it is also referred as the irradiation breeding. Irradiation breeding means what? The creation of mutations by subjecting the plant to the ionizing radiations, and the production of new variety is called as the irradiation breeding. Okay, creation of mutations by subjecting the plant to the ionizing radiation and producing the new varieties, it is called as the irradiation breeding. Here the radiations are of two type, ionizing and the non-ionizing. In the ionizing, you can see the alpha rays, beta rays, x-ray, gamma rays, and the neutrons. These are ionizing radiations, that means they produces the ions. Okay, and the non-ionizing, the only one here, the ultraviolet rays, these are included into the non-ionizing radiations. And here in the B, the different types of the chemicals are used as the mutagen to induce the mutation in the plants in the mutation breeding technique.